Over the centuries of human history, pathogen microorganisms have caused major medical problems, including epidemics and pandemics. For a long time, it was not known what caused such major medical problems because even the existence of microorganisms was not known. Advances in instrumentation, specifically microscopes, provided visualization of microorganisms and enabled research towards killing such pathogens and for treating affected patients. An example of pertinent development is, of course, the discovery of antibiotics. What did people do in the past and before the quote-unquote age of antibiotics? In this respect, there are anecdotal cases of successful elimination of pathogens from sources such as contaminated water supplies, which have the potential for affecting entire communities and cause major medical problems leading to patient death. One case was published in Science, a highly reputable scientific journal, a few years ago. The case reported in Science was an example of ancient traditional disinfection, and it was entitled Life-Saving Pitchers. In some part of rural India, people believed that storing water for drinking purposes in brass pitchers prevented illness. Scientists from a university in the United Kingdom, in collaboration with colleagues from a university in India, decided to do an experiment and scientifically determine what was the reason for such an desirable outcome. So, the experiment they did was to take some of the brass pitchers which people in rural India used and some other water containers made out of earthenware or plastic. They had two series of experiments. In one case, the pitchers were filled with sterile water, but on purpose, the researchers put in that particular containers Escherichia coli, which is a pathogenic microorganism, and they had another series of pitchers, brass, earthenware, plastic, which now contain contaminated river water from the rivers and the water sources that the people in rural area use for the drinking water. They noticed the following results. The bacteria counts dropped from as high as 1 million per milliliter to 0 after 2 days. This result was observed only in the brass pitchers. The bacterial levels in the earthenware and plastic pitchers stayed very, very high during the same time period. So, the explanation provided by the researchers was that the brass pitchers overnight released ions of copper. Copper is known for its killing capability of all kinds of cells, including bacteria. This was scientifically then the outcome obtained and the good explanation of why the brass pitchers used over centuries in rural India kill the bacteria from the contaminated rivers and protected the people of rural India. So the researchers concluded that the results support an ancient anecdotal kind of belief. I should add at this point that the good result of the ions of copper killing the pathogenic microorganism was a result of a very low concentration of the release ions. Copper ions are known, as I said earlier, to kill cells, all kinds of cells, mammalian cells, cells from tissues, bacteria. However, in high concentrations, they can be very toxic. That is the reason why any metal containing copper components cannot be used as an implant in the human body, because if the copper ions are released, especially in high concentrations, they can cause major medical problems in the recipient patients. This is an aspect which we need to keep in mind when metals are used as implantable materials for devices which are introduced in the human body. The need to prevent and fight infection resulted in developments regarding sterile conditions and sterilization of implants. This is the topic of lecture number 23. The developments discussed in the present lecture resulted from a major medical requirement, specifically that materials and devices implanted into the human body and the bodies of animals must be and remain sterile. Otherwise, if infection starts around the implanted materials and devices, that condition may cause serious illness, even leading to the death of recipient patients. These, of course, are situations which need to be avoided. We'll start with the pertinent definition. Sterile is a condition characterized by the absence of all living organisms, such as bacteria, yeast, mold, and viruses. An important clarification must be made. Sterility of implants is not the same as cleanliness. The rest of the lecture will hopefully define all aspects which are pertinent to sterility and point out the difference of sterility versus cleanliness. The pertinent definitions continue, and first and foremost is that of bioburden. Bioburden refers to the number of viable microorganisms on an implant material device prior to sterilization. Once the sterilization process is completed, there is a criterion to determine that sterility has been achieved, and that is the so-called sterility assurance level, abbreviated SAL. Sterility assurance level is the probability that a given implant will remain non-sterile following exposure to a given sterilization process. Experiments and pertinent tests are needed in order to establish the sterility of materials and devices. A test which can be conducted is the following: to take specimens of a material under consideration and take small samples and immerse them into a clear sterile liquid which has the capability to promote culture of cells. therefore should be an appropriate so-called culture medium. Allow this particular now containers in which the specimens are immersed to stay for a number of hours, let's say 24 hours, in an environment that simulates the body temperature, 37 degrees centigrade. At the end of that particular time, what one checks for is turbidity. If there is microbial growth, the original clear liquid will become turbid. And this is the evidence that the original specimen had been contaminated by microorganisms. The pathogens collected from the test just described can be further examined to identify the type. For this purpose, microbiology techniques are used in specializing laboratories, and they identify the pathogens which may be involved in subsequent infections if the material tested carries such a pathogen. Another important aspect that must be determined when sterilizing implants is the duration of exposure to sterilization in order to achieve the desirable outcome, specifically elimination of pathogens on the tested implants. For this purpose, the recommended procedure is as follows. Start by determining the bioburden on several material samples, for example, 10 to 30 sample specimens. Perform fractional run sterilization experiments. These experiments expose a smaller number of the specimens separately to the tested sterilization methods for various periods of time. After exposure to the specific sterilization process, rinse, shake, or sonicate the adhering microorganisms of some of the tested biomaterial samples. Collect all these microorganisms into a small volume of a sterile recovery fluid, such, for example, an appropriate culture medium, and determine the number of viable microorganisms remaining after sterilization for a specific period of time. At that point, now, plot the data on graphs like figure 3143, which is shown on the next slide. This graph is used to determine the effective duration of material exposure to sterilization. It is a busy graph. Let us focus on the important aspects. First and foremost, the y-axis of this graph is a log plot of the number of microorganisms as a function. Therefore, the x-axis is the duration, the time interval of the exposure of the particular pathogens to the sterilization method. Let's say that we start with a particular bioburden in the order of 10 to the 6th, or somewhere between 10 to the 5th and 10 to the 6th pathogen microorganisms. As these particular specimens carrying the pathogens are exposed to sterilization, and then we determine the number of viable microorganisms, we find that there are fewer and fewer cells which are viable, and if we plot those points, we get the straight line which is cutting diagonally on this particular graph. When that particular line cuts the x-axis at the 10 minus 6 point, which is the sterility assurance level, that particular time point is the length of time needed in order to expose a particular specimen to sterilization, the process under consideration. and this particular time point is expressed in minutes. Summarizing the results of this procedure, a reminder that the log of the number of microorganisms is plotted versus the duration of exposure to the sterilization process. The intersection of the resulting straight line with the x-axis gives us the number of minutes of exposure to the tested sterilization process, and this particular time interval is needed to achieve successful sterilization of the tested biomaterial. Another important consideration is the question which specific sterilization method to use in order to be successful in achieving the desirable outcome. First and foremost, the integrity of the implant material and device must be maintained following the sterilization method. The integrity of packages or any other enclosures in which the implant is contained and transported from the factory to the clinics and hospitals, that particular integrity must be maintained. The sterility assurance level must be maintained at all stages after sterilization, all stages of handling, transporting, storing the implants until these implants, these products, are used in surgical procedures in hospitals and clinics. 3 sterilization methods are currently used for materials and devices. Those are steam sterilization or autoclaving, ethylene oxide sterilization, and sterilization via radiation from various isotope sources. We will review briefly each one of these 3 methods of sterilization. Steam sterilization or autoclaving is a widely used method. The procedure exposes implant materials and devices to saturated steam at 121 degrees centigrade under pressure for about 15 to 30 minutes or until all surfaces of the implant reaches a temperature of at least 121 degrees centigrade. Please keep in mind that water boils at 100 degrees centigrade. Therefore, the materials and devices are exposed to temperatures which are above that of boiling water. The process of steam sterilization or autoclaving requires a special sterilization chamber, an instrument which is put together in such a way that can withstand high temperature and high pressure conditions, and these particular instruments can be found in laboratories in various sizes. We have, for example, in my laboratory a particular autoclaving unit which can accommodate perhaps a couple of bottles, 500 ml each. In addition, the department has a facility which is almost a section of a room. In big hospitals where a lot of materials and devices need to be autoclaved on a daily basis, sometimes those institutions have laboratory facilities for autoclaving which are as big as a lecture room or a laboratory. The effectiveness of autoclaving is a result of biological effects. By exposing the microorganism to the high temperature and pressure, that is a process which kills them because it destroys their metabolic and structural components which are needed for proliferation. One of the underlying mechanisms involves coagulation of essential enzymes. Another one is disruption of protein and lipid complexes, etc. Autoclaving is a sterilization technique which is currently used in laboratories, research laboratories, and it is used for sterilizing glass, metals, and liquids. It is used also in hospitals for sterilizing metallic surgical instruments, heat-resistant surgical supplies as linen drapes, dressings, etc. The advantages, which explains the popularity of this method, is that it's a relatively simple method. It is fast, it is easy to control, it is efficacious, that means it gives us a good result, the desirable result, and it doesn't leave any toxic residue. Disadvantages of this sterilization method is the combination of the required high temperature and pressure conditions, because these conditions are the ones which metals, for example, and packaging of, let's say, that involves linen and drapes, etc., can withstand the high pressure and temperature condition. However, polymeric materials cannot be sterilized using this method because most of the polymers melt under high temperature conditions. Currently, many of the materials and devices which either come in contact with the human body or are introduced into the human body are composed of polymers. These materials cannot be sterilized using autoclaving because they melt. Therefore, other procedures are needed to specifically sterilize polymeric materials. One of the methods used is ethylene oxide sterilization. The ethylene oxide is usually abbreviated capital letter E, small case t, capital letter O, ETO. During the process of ethylene oxide sterilization, implant biomaterials and devices are exposed to ethylene oxide gas, concentration 600 to 1200 milligrams per liter, in a relative humidity of 40 to 90%, at a temperature of 30 to 50 degrees centigrade. Keep in mind, body temperature 37 degrees centigrade. This process must take place inside a very special sterilization chamber and facility for 2 to 28 hours. The special sterilization chamber is needed, and in order to have the air or the gas removed from that particular chamber at the end of the sterilization process, the environment with the gas is established initially by injecting the gas into the particular chamber, and it is built in such a way that the environment of the ETO gas is maintained for the duration of the sterilization procedure without any unwanted leaks. And at the end of the procedure, any ethylene oxide gas which must remain inside the chamber must be removed very carefully and without any complications, leaks, and problems. Ethylene oxide is a very effective sterilization method because it affects microorganisms at the molecular level, at the nucleic acids, RNA and DNA, and therefore it is lethal to microorganisms. However, this particular method of sterilization requires special considerations for the following reasons. ETO is a toxic chemical and a suspected carcinogen. Both pure ethylene oxide gas and mixtures without a proven inert compound are flammable and potentially explosive. However, this particular technique of sterilization is used currently for a variety of applications: surgical sutures, intraocular lenses, ligament and tendon repair devices, adsorbable and non-adsorbable meshes, neurosurgery devices, adsorbable bone repair devices, heart valves, vascular grafts, etc. A lot of these particular implant devices are of course composed of polymers. The ethylene oxide sterilization method has several advantages. First and foremost, it has high penetration ability. This capability assures that all facets of a three-dimensional structure of the various implantable devices, all facets are sterilized. It is a very effective method, even at low temperatures. Remember, it works at temperatures between 30 and 50 degrees centigrade. It is compatible with many types of materials, including polymers, which are very sensitive to temperature ranges because they melt at high temperatures. The sterilization method of ethylene oxide, however, has certain disadvantages. And first and foremost is the fact that this particular gas is toxic and it is potentially causing cancer. And this can be the result of even a few residue molecules of the particular gas on materials and devices. This particular residue remains clinging on the surfaces of the materials and devices even after the process of sterilization is completed. For this reason, extra self-time is needed to remove effectively the clinging residue before the materials and devices are delivered to hospitals and implanted. The self-time is required and it must be taken into consideration when biomedical engineers who are in charge of this particular part of the operation are responsible for delivering to a particular client, a particular hospital, the requested implant devices. They need to take into account that extra time is needed to remove all clinging ethylene oxide molecules from the devices. In the past, there were some unfortunate incidents because the removal was not complete and implants with ethylene oxide residue were implanted and caused problems in patients. In addition to the effects of ethylene oxide molecules to humans and various patients, the molecules are detrimental to the personnel who work in the facility which uses ethylene oxide for sterilization purposes. Last but not least, ethylene oxide at the end of the procedure cannot be released from the special chambers and facility into the atmosphere and the environment because of the potential toxicity and carcinogenicity of this particular chemical compound. Another method currently used for sterilizing devices and biomaterials is the use of a radioactive isotope such as cobalt-60 for sterilization purposes. Radiation sterilization involves exposure of implant materials and devices to gamma rays released from isotopes such as cobalt-60 at a dose of 25 kilograys. The use of radiation sterilization requires special irradiator machine and a facility to store and use any radioisotope. The biological effects of this sterilization process are well documented. Gamma rays have high penetrating ability, but only in the linear mode. The gamma rays ionize key cellular components such as the nucleic acids and therefore kill microorganisms. Currently, radiation is used to sterilize various biomedical materials and devices such as surgical sutures, drapes, metallic bone implants, knee and hip prostheses, syringes, neurosurgery devices, etc. The advantage of this particular method is that it is simple, fast, and effective. It is compatible with some, but not with all polymers. And as biomaterials engineers, we must be aware of the polymers which are not compatible with this particular method. Another advantage of radiation as a sterilization method is the fact that it doesn't leave any toxic residue. As with the other two sterilization methods discussed, radiation has certain disadvantages. First and foremost is the high cost required for building a special facility and installing the isotope and running that particular facility for sterilization purposes. Due to the continuous decay of any isotope that may be used for the sterilization purposes, these isotopes must be replaced at regular time intervals. In addition to the appropriate funds and expenditures to buy and bring in a new isotope, one must take into consideration the downtime. During that particular time period, the facility is closed and there are no sterilization processes taking place. Therefore, either the procedure for sterilizing certain products may be delayed, or if the facility is run for other organizations or other companies, then there is not going to be any revenue during the downtime when the operation of the facility is down and the facility is closed. One other aspect that must be kept in mind is that radiation sterilization is not compatible with certain polymers. Biomedical engineers must be aware of that particular aspect and must make sure that those polymers are not exposed to radiation sterilization. What happens that is detrimental is that exposure to radiation breaks down the backbone of polymers and therefore changes not only the chemical composition but then the function of the particular device which is composed of these particular polymers. And one may be under the impression that certain chemical compound, a certain polymer is the one out of which the device is composed and therefore will function, and the actual performance inside the human body might be totally different. Last but not least, unsafe radiation facilities are environments in which the personnel working in those facilities are exposed to health-related hazards. This is an aspect which needs to be addressed and eliminated in order to have a safe facility for the purpose of radiation sterilization of biomedical materials and devices. In addition to the three widely used methods of sterilization of materials and devices, there are several other emerging technologies which are being used and experimented with. For example, gaseous chlorine dioxide. This is another gas which has been experimented with for the purposes of sterilization. Low temperature gas plasma. Plasma here, it's not the liquid part of blood, but it is a gaseous environment containing ionized gases. Gaseous ozone. Ozone is known to have oxygen radicals, which of course kill microorganisms. A vapor phase hydrogen peroxide. This is a very interesting method, and I would like to relate to you a particular case which happened in the building in which the biomedical engineering department is housed at UTSA. When we moved here in this building, in the AT building in 2009-2010, we had problems with contamination of some of the cell culture rooms. And in order to eliminate all possibilities of microorganisms which had invaded the labs and they were therefore were causing all kinds of problems with the experimental studies conducted in those laboratories, a particular company in town was hired, and what they did is to bring up a machine which produced gaseous hydrogen peroxide, put a big hose and brought that particular gaseous hydrogen peroxide into the laboratory. We had to open all doors and all cabinets, etc., and the atmosphere of the laboratories was penetrated by hydrogen peroxide for a number of hours, and after that the laboratories were disinfected. It is a very effective technique of sterilizing medical implants, including whole rooms, whole laboratories, as I told you, and it is a relatively new or emerging technology. Machine-generated X-rays is another emerging technology, and a few others which are which appear in the press from time to time. Sterilization of implant materials and devices is a responsibility of biomedical engineers working in pertinent industries. The products delivered to hospitals and clinics from the companies from industry must be sterile before they are packaged and must remain sterile during handling, transport, and delivery to the destination hospital and clinic. How are the sterile conditions maintained afterwards? These aspects are the responsibility of healthcare colleagues, the responsibility of the hospitals, clinics, surgery rooms, and intensive care units where these products will be used in conjunction with patients. A brief review of the sterile conditions and practices used in surgery rooms and hospitals follows. The sterile conditions used in clinics and surgery rooms are as follows. Following delivery of sterile implant materials and devices to the hospitals and clinics, and if it's needed, these materials and devices must be stored there under sterile conditions. The surgeons and all assistants in the surgery room wear sterile gowns, masks, gloves, head covers, shoe covers, etc. The surgery rooms are built with special ventilation schemes. Specifically, the air blown in the room is blown away from the surgical bed on which the patient lies. Therefore, the possibility that the ventilation from the building will direct the ventilation air on the patient, therefore bring pathogens, is eliminated by using this special ventilation scheme in surgical rooms. The temperature in the operating rooms is kept lower than in the other hospital rooms because it's known that the pathogen microorganisms and they grow much faster at higher temperatures. The surgery rooms of the hospitals are built using special non-porous materials which can be sterilized using appropriate solutions of chemicals at regular time intervals. The surgical beds and the other items in the operating rooms are made of materials which can be sterilized at regular time intervals. In addition, before surgery, all medical personnel participating in the procedure scrub. That means that they have, without touching faucets, etc., they have to, first of all, wash thoroughly using appropriate soaps their arms and hands, and then at the very end treat their skin of their hands and arms with appropriate chemical solutions which are known to kill microorganisms. Before surgery, the skin of the patient at the anatomical site where surgery will take place is prepped. That means that it's sterilized using chemical solutions which are known to kill microorganisms. These solutions are applied on the skin of the patient. Only sterile instruments are used during the operation surgery. All surgery instruments are used once and for only one patient. Before as well as after surgery, the patients are given pertinent antibiotics to prevent infection and related health complications. These procedures used to promote and maintain sterile conditions are hopefully helping the situation, and if not 100% preventing, still they're eliminating and decrease satisfactorily the potential for infection of patients in hospitals and clinics. This is the end of lecture 23, Sterile Conditions and Sterilization of Implants.